Welcome everyone, or welcome back if you are with us for our first session today. As you know, this is the Current Directions in Psychological Science speaker series presented by Pearson and APS. Without further ado, I'd love to introduce our next speaker, Christine Wagner from the University of Albany. Dr. Wagner's research examines the role of progesterone in neural development, specifically the role of progesterone and its receptor play in the development of sex differences in the brain, and in the development of the mesocortical dopamine pathway and cognitive behaviors. She has received grants from the National Science Foundation, as well as from the National Institutes of Health and March of Dimes Foundation for Birth Defects. Dr. Wagner has served as expert consultant for the World Health Organization and Centers for Disease Control. And her talk today will focus on sex differences in brain and behavior.
My pleasure to speak with all of you today, and I'm going to tell you a little bit about research in behavioral neuroscience and how it might inform the study of psychology in the classroom. So as we all know, of course, psychology is the study of human behavior, and because all behavior at some level is a reflection of brain activity, the field of behavioral neuroscience investigates how brain function regulates normal behavior. But in addition, behavioral neuroscience also examines how changes in brain function might produce or maintain behavioral dysfunction. Now, because humans come in two flavors, male and female, one of the fundamental questions in psychology is whether there are sex differences in human behavior. And if so, do these sex differences have a biological basis? In other words, are sex differences in behavior the result of sex differences in the brain? And so we all likely have anecdotal evidence suggesting that, yes, indeed, there are sex differences in behavior. We think that men and women think differently and that boys seem to act differently from girls on the playground. But perhaps more convincing evidence is that there are significant sex differences in the incidence of behavioral disorders. So this slide shows the incidence of a variety of psychiatric and neurological disorders in males and females, and very creatively, males are represented in blue and females in pink. And several disorders, particularly attention deficit disorders and autism, are diagnosed much more often in males than in females. And in fact, the ratio is about 8 males for every 2 females diagnosed with these disorders. Now, on the other side of the coin, some disorders are much more prevalent in females. About 70 to 80% of eating disorders and depression and anxiety disorders are diagnosed in women. And interestingly, if we look at when these disorders are typically diagnosed, we see that the disorders that are more prevalent in males are those typically diagnosed in childhood, whereas those disorders more often diagnosed in adulthood are more prevalent in females. Now, what this suggests is that the susceptibility of males to some disorders may have a developmental and perhaps biological basis. And so examining sex differences in behavior has significant implications for psychology in general, but more specifically may inform how we understand behavioral disorders. So the question then becomes, are sex differences in human behavior the result of sex differences in biology, or are they simply the consequence of social and cultural influences? And of course, this raises the age-old nature versus nurture question. So sometimes it's really easy to parse out the role of nature and the role of the environment. For example, generally speaking, there are sex differences in hairstyle. And it's easy to see how this sex difference is almost certainly the result of social influence and cultural expectations. And so nurture has the upper hand. But there are also sex differences in snoring. Men snore more than women. And this is easily explained by sex differences in anatomy and physiology. And so nature trumps nurture. However, there are sex differences in crying behavior. Girls tend to cry more than boys. Women tend to cry more than men. And the roles of nature and nurture in this case become a lot less clear. So do girls cry more because society teaches boys that crying is not manly? Or are sex differences in crying the result of biological differences in emotionality? So in these more complex cases, it can be really difficult to determine the biological basis for sex differences in behavior. And so scientists have approached this problem in at least two ways. So first, the use of animal models has been extremely useful. So for example, using laboratory rodents, scientists can examine sex differences in the brain and how those differences in brain correlate with sex differences in behavior. And another useful model has been to examine naturally occurring conditions in humans. So behavior can be observed in individuals in whom a medical condition has altered their physiology, but their environment remains the same. So nature is altered while nurture is held constant. So how do biologically based sex differences arise in behavior? Well, research from animals has clearly shown us that males and females are differentially exposed to steroid hormones during development. And this produces changes in neural development, which then in turn produce permanent differences in brain structure and function. And then changes in brain function lead to long-lasting, sometimes permanent, and often very dramatic sex differences in adult behavior. So to explain that a little further, in laboratory rodents, it's been clearly established that sex differences in behavior are the result of differential exposure to the steroid hormone testosterone during brain development. So in early fetal life, the brain is actually undifferentiated, meaning it's neither male nor female. And rather, the brain remains bipotential, meaning it has the capacity to develop either in a male-like pattern or in a female-like pattern. However, the fetal male testes begins to secrete the androgen testosterone before birth. And in contrast, the female ovaries don't begin to secrete hormones until well after birth. Exposure of the brain to testosterone in the male then causes the brain to develop in a male-like pattern, and this produces male typical behaviors in adulthood. And then in the absence of testosterone in the female, the brain develops in a female-like pattern, we think by default, and this produces female typical behaviors in adulthood. Now, there are some very classic studies done in the 1970s that clearly demonstrate the role of testosterone exposure in development on adult behavior. So in adult rodents, there is a very clear sex difference in the incidence of aggressive behaviors. Males will demonstrate high levels of aggression toward other males, whereas females will rarely fight. And in one very early study by Peters et al., male rodents were castrated at various times during development, thereby removing the source of testosterone. So when the rodents reached adulthood, testosterone injections were given to produce normal adult levels of testosterone. And then aggressive behavior toward another male was measured in adulthood. And so this graph on the left shows typical levels of aggression in adulthood in normal control males. And you can see aggressive levels are pretty high. Castration on the day of birth or on postnatal day 2 significantly reduced the levels of aggression in adulthood. However, castration on postnatal day 6 or 40 were much less effective at reducing aggression. And so these findings suggested that testosterone during the first few days of life are critical for the display of aggression in adulthood. And in another classic study shown on the right, females were administered testosterone during development, and aggressive behavior toward a male was examined in adulthood. And as seen in this graph, the level of aggression in normal control females is very low. However, if females were given testosterone injections on the day of birth, their levels of aggression were significantly increased in adulthood. Testosterone injections on postnatal day 6 or postnatal day 24 were much less effective. And these studies, and many more like them over the decades, have demonstrated that exposure to testosterone during neural development is necessary and sufficient for later aggressive behavior. And so these studies also suggest that there's a sensitive period during neural development. Testosterone exposure during a specific developmental window is most effective at altering aggressive behavior. And testosterone either before or after this window has very little effect on later behavior. So these studies suggest that testosterone is altering neural development in some way that is so significant that behavior is altered months after the testosterone exposure. And so what does testosterone do in the developing brain? Now, to answer that question, it's first important to understand a little bit about how steroid hormones like testosterone act within cells of the body. So testosterone, like other steroid hormones, can readily enter the brain from the bloodstream. And once in the brain, steroid hormones interact with receptors that are typically located in the nucleus of the cell. These receptors become activated by the steroid hormone and then can interact with the cell's DNA. Here, the receptors either enhance or suppress the expression of specific genes, and this, of course, can exert very profound effects on fundamental processes of neural development. So in this way, differential exposure of males and females to the steroid hormone testosterone can result in very different patterns of development in the brain. So for example, testosterone could alter the number of specific cell types in the brain. It could also create sex differences in neural connections. And testosterone can even produce sex differences in the sensitivity of the brain to yet other steroid hormones. And then in the next few slides, I'm going to show you just a few examples of the way in which testosterone can alter neural development and show you some results from some experiments in which steroid hormones produce sex differences. So in the adult rodent brain, there is a sex difference in the number of dopamine cells in the hypothalamus. So in this example, females have more dopamine cells than males do, as seen as these black dots in these photographs. Now in this experiment shown in the graph on the right, females were given injections of testosterone either on postnatal day 4 or postnatal day 10, or they received control injections. And the number of dopamine cells was then counted in adulthood. Now testosterone exposure during development significantly reduced the number of dopamine cells. And in fact, testosterone in treated females had dopamine cell numbers that were similar to that seen in males. And so these findings suggest that the sex difference in dopamine cell number was the result of differential exposure to testosterone during development. Now, there are also sex differences in the connections between brain regions in the adult brain. So in other words, there are sex differences in the way in which neural signals are sent within the brain. Now, in this example, there are very strong connections from region A to region B in males and a very weak connection in females. And this can be seen in these photographs as a sex difference in the density of these white fibers in region B that originated from region A. And in this very elegant experiment, region A and region B were removed from fetal animals and were placed near each other in a petri dish in culture. And the growth of fibers from region A toward region B in the petri dish could then be observed using special fluorescent stains. So in the first panel here, if region B came from a male, there were many fibers that extended from region A to region B. But in the second panel, if region B came from a female, then very few fibers extended from region A to region B. However, as shown in the third panel here, if region B came from a female that had been treated with testosterone, the fibers growing from region A to region B looked just like a male, no matter the sex of origin of region A. And so these findings suggest that testosterone is acting at the target for growing neural connections, and that differential exposure of males and females to testosterone during the formation of these connections could produce sex differences in the way the brain is wired, so to speak. And this, of course, could have profound effects on brain function later in life. Now, the last example is work from my laboratory, where we've shown that exposure to testosterone can alter the sensitivity of the developing brain to yet other steroid hormones. So as I mentioned earlier, steroid hormones activate nuclear receptors, which in turn alter the expression of specific genes. And my lab has demonstrated that exposure to testosterone can increase the expression of receptors for other steroid hormones during neural development. So this slide shows the expression of receptors for progesterone, another steroid hormone, in the hypothalamus of male and females on the day of birth. Males express very high levels of progesterone receptor, whereas in females, progesterone receptor is virtually absent. And we've conducted numerous experiments over the past decade demonstrating that this sex difference is the result of testosterone exposure in males, increasing the expression of the progesterone receptor gene. Now, these findings suggest that the male brain may be more sensitive to progesterone than the female brain during critical periods of development. It also suggests that progesterone and its receptor may play an important role in the development of sex differences in the brain. And in fact, very recent, not yet published data from my lab suggests just that. So in adult mice, there's a significant sex difference in the density of serotonin connections within the hypothalamus. Males have a very high density of serotonin fibers compared to females. Now, my lab has a strain of mice that has a mutation in the progesterone receptor gene. We call them progesterone receptor knockout mice, or PERCO mice for short. And these mice lack functional progesterone receptors, and they are, by and large, insensitive to progesterone. So when we look at the density of serotonin fibers in these progesterone receptor knockout males and females, the sex difference is abolished. Now, this suggests that progesterone and the progesterone receptor may play an important role in the development of the sex difference. And examining a role for progesterone in neural development is having increasing significant clinical implications. It's becoming increasingly common for pregnant women to be given injections of progesterone for the prevention of premature delivery. And this is being done despite the fact that there is very little known about the effects of progesterone exposure on brain development, and particularly little known on the effects of progesterone on sex differences in human brain development. So rodents make a great model to study sex differences in brain and behavior, but what about humans? Are there measurable sex differences in human behavior? And the answer is yes, and in fact, the largest and most replicable sex differences in human behavior are found in play behavior in children and spatial ability in adults. Now, there are rather large sex differences in play behavior in children, and this graph shows the percentage of time that either boys or girls spent playing with toys that are either typically masculine, like cars, trucks, and guns, shown in gray, or toys that are typically feminine, like dolls, tea sets, and makeup, shown in green. Boys spent significantly more time playing with masculine-type toys and spend very little time playing with feminine-type toys. Girls are slightly more flexible in their toy preference, but they still have a slight preference for dolls and feminine-type toys. Now, of course, it's really difficult to determine the cause of this sex difference. It could be very easily argued that this sex difference in play behavior is simply the result of the influence of a society that teaches children what toys they should play with. Girls are handed dolls, boys are handed cars. Or perhaps it's the result of marketing by toy companies that push certain toys on each gender. On the other hand, perhaps the sex difference in toy preference is a biological one and the result of sex differences in the brain. So if this type of sex difference were to exist in non-humans, it would provide pretty strong argument for a biological basis in the sex difference in this behavior. And now in this very fascinating study, toy preference was examined in monkeys. Now the monkeys in this study had never been exposed to toys prior to the study. And in addition, they had certainly never been exposed to marketing or a gender-biased society. And so this graph on the right shows the number of interactions that male and female monkeys had with either a toy truck, shown in gray, or a plush doll, shown in green. Now, male monkeys interacted significantly more with the truck than with the doll. And in fact, males were observed to move the truck along the ground just as a child might do, playing with a truck. Now, more similar to humans, female monkeys showed more equal preference for the doll and the truck. But female monkeys were observed interacting with the doll in ways that were similar to the way mother monkeys interact with their infants. And since it's hard to argue that sex differences in toy preference in monkeys are the result of environment, these findings strongly suggest that the sex differences in play behavior probably arise from a biological difference in the brain of males and females. Now, one of the other largest measurable sex differences in human behavior is found in spatial reasoning. Now, spatial reasoning is the ability to reason about the position or the movement of objects in space. And as an example here, this is an example of a spatial reasoning task on the left. In this task, subjects have to determine whether the object on the left is the same or different from the object on the right. And this task requires subjects to mentally rotate the objects in space. And on average, men score about 35 to 40% higher on such tasks compared to women. And if you're playing along at home, the two objects in A and B are the same, and the two objects in C are different. Now, again, it's really difficult to determine if this sex difference is simply represents the influence of education and society, for example. Perhaps boys are encouraged to excel in these kind of spatial tasks, whereas girls are encouraged in other types of tasks, like verbal tasks. Or perhaps the sex difference reflects differences in brain function. Now, to address this question, scientists have turned to a naturally occurring endocrine condition in humans. So, for example, there is a condition in humans called congenital adrenal hyperplasia, or CAH. And this condition is caused by mutation in an enzyme that synthesizes hormones in the adrenal gland. And the result is that the adrenal gland secretes abnormally high levels of androgens during fetal life. And so the condition is typically corrected at birth with the administration of medication. Now, this CAH condition then produces human females that have been exposed to levels of testosterone before birth that are similar to that normally seen in males. However, they are then raised as typical girls. And so the behavior of these CAH girls can then be compared to their unaffected sisters, and it creates a really elegant separation of nature and nurture in humans. Now, a recent study examined spatial ability in women with CAH. So in this study, subjects had to perform a virtual 3D maze in which they had to find a target as they moved through virtual space on a computer. And this graph shows the latency to solve the maze. For control males, shown in blue, control females, shown in pink, and CAH females, shown in green, that had been exposed to high levels of androgen before birth, but raised as girls. Now, there was the expected sex difference in spatial ability, in which males solved the maze more quickly than females did. However, CAH females solved the maze in a time that was similar to males and significantly faster than control females. So these findings are consistent with the idea that exposure to androgens before birth, exposure to testosterone before birth in humans, creates sex differences in spatial reasoning later in life. Now, the CAH condition can also be used to examine the sex difference in toy preference. Boys spent more time than girls playing with toys like cars, trucks, and guns, and girls preferred to play with dolls, tea sets, and makeup more than boys. However, CAH girls were more similar to boys in their toy preference. CAH girls spent more time playing with the boys' toys than control girls. The CAH girls have been raised with all the social biases of control girls, and yet their play behavior is more male-like. And these findings suggest that sex differences in toy preference in children may also be the result, at least in part, of testosterone exposure during neural development. So in summary, animal models reveal that males and females are differentially exposed to the androgen testosterone during development, and that testosterone acts on the brain to alter fundamental processes of neural development. Now, this undoubtedly leads to significant sex differences in the way the brain functions. Differential exposure to testosterone during critical windows of neural development also results in permanent sex differences in adult behavior. Now, of course, in humans, the story is much more complex and more difficult to empirically test the role of hormones in behavior. But CAH girls are more male-like in their spatial ability and play behavior despite being raised as girls. And this suggests that exposure to androgens before birth in humans plays at least some role in sex differences in behavior. Now, future research in this area of behavioral neuroscience will undoubtedly reveal that sex differences in behavior are the result of interactions between several factors, one of which is steroid hormones. And a paper recently published in Nature Neuroscience presents a new framework for the development of sex differences in brain and behavior. And this paper suggests that at the genetic level, genes found on sex chromosomes triggered gender-specific effects in developing brain. Then these effects interact with the differential production of steroid hormones in males and females. And then in addition, gender-specific interactions with the environment may cue further sex differences in neural development and may even differentially alter the biology of males and females. So what implications do these findings have for the field of psychology? So first, understanding how sex differences in neural development influence sex differences in later behavior can contribute a lot to our understanding of human behavior in general. But furthermore, the investigation of biological sex differences in behavior can have significant implications for understanding the vulnerability of males and females to specific behavioral disorders. In addition, this type of research may also shed light on why some disorders present very differently in males and females. So for example, these photographs represent a very typical sex difference in the way attention deficit disorders emerge in the classroom in boys and girls. So understanding that the brain works differently in males and females could inform diagnosis and intervention in these kinds of cases. So in closing, I guess the take-home message is that in the study of human behavior, we must include an appreciation of sex differences in biology and in the brain and in behavior. And thank you very much.
We'll now turn it over to Master Teacher Marc Tetel for our interactive Q&A. Dr. Tetel is a professor at Wellesley College in Massachusetts and is a member of both the neuroscience program as well as the biological chemistry program at that school. So, hi Marc, how are you?
Hello, good, how are you?
Good. So similar with our other sessions, we have some questions that came in ahead of time. We also welcome any questions that people want to submit using the questions chat feature. So we'll go ahead and get started. Our first question for you, and these are all related to teaching the psychology class or biopsychology class. But the first question, I teach a large lecture hall with 100 plus students and would love suggestions for how to keep my students engaged and participating during lecture. What suggestions do you have?
Yeah, so that's a great question and a tough one to address, I know. First, I just want to thank Chris Wagner for a wonderful talk. I thought that was great. I think just as Chris did in her talk, obviously one of the most important things is to keep the material accessible and, to build up a foundation. I think with college students in general, anytime you can relate the topics to them, it keeps them more engaged. And so I think with these issues of sex differences and how biology can influence behavior, I think connecting to students and what they're interested in is important. So I think if someone were to start a lecture on this kind of topic, one thing that students are really interested in is dating. And if you're talking about hormones, well, there's lots of recent data that has shown that hormones influence things like decision making. So studies have shown that women across their menstrual cycle will find different types of faces attractive. So at one part of their cycle, they'll find masculine faces more attractive, and another part of their cycle they'll find other types of male faces attractive. So I think keeping topics related to being able to relate it to the students is really critical. And then I think as far as keeping them engaged, I think in a large lecture hall, I guess asking open-ended questions is a good way to do it. So asking students to design experiments where you can talk through the different experiments, asking students their opinions about these controls, some controls compared to other controls, is a good way to keep them engaged. And then one thing I always do when I start a sex differences lecture is I put up on the board, I put the male symbol on one side of the board and the female symbol on the other side of the board, and then I just have students start to list sex differences and make a list of things that are different between the sexes, putting the differences under those symbols. And I think that's a way to sort of get students engaged in thinking about differences. And if the class just puts up behavioral differences, then you can point to the fact that there are also physical differences or the reverse.
Thanks. So we have a question that just came in that says, my students have a difficult time understanding the difference between sex and gender. Any ideas to help them understand this difference?
Well, I think so. Sex would be the male or female connotation to an XY would be a male and XX would be a female, and gender has to do with issues of identity. So there's really that gray area in between.
And any suggestions, activities in class?
There are issues of sexual identity and along those lines. Pardon me?
No, that was great. And then the question also was, do you have suggestions for in class how you can help them understand this concept if they have trouble?
I think just explaining those differences. There's sort of a biological difference between males and females, but then there are identity issues that can be influenced by biology, but also involve identity issues as well.
Okay, thanks. So we've got a couple student questions. This first question says, I'm currently a student in a master's program for professional counseling. How can I utilize this knowledge when I'm counseling the different sexes?
Counseling, pardon me, that?
When I'm counseling the different sexes, so when I'm counseling a male versus a female.
Oh, well, I think it's just important to understand, as Chris Wagner was talking about in her seminar, that there are ideas that there are biological foundations behind these differences. So I think it's important to be aware of those and to point them out to patients as one's counseling them, but also to understand that there are individual differences. So while Chris pointed out some studies that were done that showed differences between sexes, that doesn't also mean that there are a lot of variation between individuals. And so I think obviously a counselor would have to be very sensitive to that, understanding that in the general there are differences, but there's a wide range, especially when we're talking about human sex differences, large range within individuals.
Okay, great. And here's a good one. So for undergraduate psychology classes, when you discuss neural differences between men and women, how do you prevent them, so students, from becoming a justification for discrimination?
How do we prevent them from, how do we prevent the differences?
When you talk about the differences, yeah, how do you prevent that from becoming a justification for discrimination amongst the students in the classroom?
Well, I think the point to remember is that while we know about sex differences, say, in the brain, and we know that there are differences in brain structures, that doesn't necessarily, we don't really know the function of those differences. And I think that's always something that's important to remember. So while we may know that brain region A is different in males than it is in females, we don't really know what that difference causes or what that difference elicits. And so I think it's very dangerous to then make that jump to assume that because these differences exist, they may have concrete effects. And I think these also become important in issues like sexual identity and sexual preference. So we know there are differences in the brain between homosexual men and heterosexual men, but we really don't know what the implications of those differences are. And so it's important to remind ourselves that we don't understand those differences. We just know the differences exist.
Mm-hmm. Thanks. And so our next question is, I focus my course more on where the science of brain and behavior is going, not so much where it has been. Do you have suggestions for how to best keep up with this current research, so specific websites or news feeds, things like that?
Sure, yeah, there are a lot of wonderful websites. I think anyone who's involved in undergraduate behavioral neuroscience research would want to check out the website from Faculty of Undergraduate Neuroscience. This is a website devoted to undergraduate neuroscience education, and they provide educational workshops. They have discussion pages. They actually have a journal, Journal of Undergraduate Neuroscience Education, that gives ideas for labs and courses and teaching materials and pedagogy. And that's at www.fundfaculty.org. And then there's the Society for Neuroscience webpage. And the Society for Neuroscience is a huge international organization with over 40,000 members. And they have an education tab that I know has information on Brain Awareness Week. So they have actually PDFs that you can download that have brain facts and brain briefs. These are two-page summaries of most of the neurological disorders and topics like sex differences in the brain. They also run a Brain Bee off that website. So this is a way to get undergraduates involved in outreach programs where undergraduates can go into high schools and instead of doing spelling bees, actually do brain bees where the students are tested on their knowledge of neuroscience. And this is a national brain bee that's held, so winners in high schools will go on to regional meetings and then they can go on to national competitions, I mean. And one way that I actually stay up on sort of current ideas and things that come up in the lay press is a colleague of mine at Michigan State University, Marc, has a daily newsletter of sort of interesting stories in behavioral neuroscience that have appeared in the lay press. And you can subscribe to this, and then every few days you get an email that sort of summarizes some of these topics. And they have ranges from, you know, Michael Jackson and how propofol works to neuroscience of addiction or maybe treatments in schizophrenia. I think the other day I saw one on altruism and research that's being done on altruism in chimps. And that's another great resource for keeping up on current ideas that are hot in the lay press.
Thanks. So our next question is, what suggestions do you have for incorporating research in behavioral neuroscience and making it relevant for the intro psychology student? So in that, you know, that class, their students may or may not be psychology majors.
Well, yeah, I think these websites offer some great ideas of how to incorporate it, and this newsletter that I just spoke of, I think, brings up interesting topics. I think also, as I started out making the research relevant to students and their age group, one assignment that I've liked to do is have students find an article in a newspaper that's interesting to them. And then find the original research article that pertains to, and then they can actually, by reading the lay press article, understand that topic, and then go deeper into the actual research article, and maybe write a brief summary of that research article. So I think that really shows to them their developing sophistication of understanding neuroscience as the course goes on. And I think doing really what Chris Wagner did in her talk, and that is, you know, bringing up an idea that seems kind of obvious, that there's differences in play behavior between boys and girls, and I think most people will accept that, but then showing them that there's a biological underpinning to this. And I think the best way to do that is to, if there's animal data that really supports that, as Chris showed with the monkey studies and differences in play behavior. I think that's one of the, in my mind, one of the more important things to get across to to people in intro psych classes, people who may or may not be psychology majors, that some of the things we take for granted, like differences in play behavior, actually have a biological underpinning.
Okay, now we have a question, another question from a student, which is, what careers are open for students who study neuroscience?
Oh, okay. Well, there are a lot of different careers for undergraduates. They can pursue careers as research technicians in research laboratories or in pharmaceutical companies. There's also positions in pharmaceutical or health product companies as marketing and sales representatives. And even in federal, state, and local public health programs, they always need people who can do health analysis. And many students in neuroscience can go on to other degrees, getting PhDs. And if someone has a PhD, then that opens up faculty positions at research universities or medical schools, dental schools, nursing schools. And then there are also research careers in as I said, pharmaceutical companies and biotech companies. Excuse me. I've had students go on to have careers in patent law. Students go on to have careers as science writers. I mean, I think the brain is such an interesting field to so many people that they always need science writers who can understand both the psychology and the biology of how the brain works. And I've even had students go on to work on Wall Street, to do stock analysis for Wall Street companies specializing in biotech firms or pharmaceutical companies. So there's really a wider range of careers that are available to either undergrads or people who want to go on to get further degrees.
Thanks, Marc. It's really interesting. So our last question, and I think you kind of touched on this with your websites earlier, but do you have additional suggestions for how students can get involved in neuroscience research?
I think that obviously if there are opportunities at their school to get involved in research, and maybe programs during the year to do research for credit, or even research in the summer where they can work full-time. I know many schools have programs set up like that. If this is something that interests students doing summer research, I would direct them to that website I spoke about earlier, the Faculty for Undergraduate Neuroscience. And they have a whole page set up. If you go to that site, and I think go under Students and Internship Opportunities, there's a long page of paid internships that are available at research universities for students from other universities and colleges. So these are usually 10-week programs where students get paid to do work and also get a housing allowance. And it lets them get involved in research and do research full-time on a topic that interests them. And these programs actually serve as recruitment tools for graduate programs. So these are researchers who are invested in recruiting undergraduates into their lab and are committed to education. So I think they're usually invested in it being a positive research experience for people. And also, if students do this and get involved in research, I would encourage them to also look at the Faculty for Undergraduate Neuroscience webpage, because they provide travel awards to attend the Society for Neuroscience meeting, which is an international meeting that's held once a year all over in different places in the United States. This organization provides travel awards for students to present their research. So I think there's a really big push to get undergraduates involved in neuroscience research and hopefully making tomorrow's neuroscientists.
Well, thank you so much. That brings us to the end of this session. And I know there are a lot of you who have questions that were not answered that we didn't get a chance to get to. So we'll respond to those by email after the event is over. But I just want to thank Dr. Wagner and Dr. Tetel for volunteering their time to share their current research and teaching strategies with all of us today. And for everyone on the line, I hope you will stay. We will be back on in 15 minutes, 2 p.m. Eastern. We'll start our next talk, which will focus on culture and human development.